Bismillahirrahmanirrahim. Plasma proteins are the proteins which are present in the blood plasma and consist mainly of the albumins and globulins, and they play a part in maintaining the osmotic pressure. Early researchers discovered the plasma proteins on account of their solubility in organic solvents by techniques like salting out, and three groups have been identified: fibrinogen, albumin, and globulins. So you can read about the plasma proteins because the early researchers discovered them. Electrophoresis yields alpha 1, alpha 2, beta, and gamma fractions of the plasma proteins. These can be drawn on a graph like this. Alpha 1 fraction contains alpha 1 antitrypsin, thyroxine binding globulin, retinol binding globulin, transcortin, and orosomucoid. Alpha 2 fraction contains alpha 2 macroglobulin, ceruloplasmin, haptoglobulin, and prothrombin, whereas beta fraction contains hemopexin, transferrin, plasminogen, and gamma fraction contains the antibodies or the immunoglobulins. That is a relative comparison between the sizes of the different plasma proteins. So that's the categorization of the plasma proteins. The aggregate concentration is around 7 to 7.5 grams per dl. Roughly 80% of the plasma proteins are synthesized in the liver. These include albumin, fibrinogen, transferrin, and most components of the complement in blood coagulation cascade, with exception of von Willebrand factor, which is synthesized in vascular endothelium. One prominent exception is gamma globulins, which are synthesized in lymphocytes. As is case for proteins destined for secretion from a cell, the genes for plasma proteins code for an amino terminal signal sequence that targets them to endoplasmic reticulum. As this leader sequence emerges from the ribosome, it binds to transmembrane protein complex in the endoplasmic reticulum called the signal recognition particle. The emerging polypeptide chain is pulled through the signal recognition particle into lumen of the endoplasmic reticulum, during which process the leader sequence is cleaved off by an associated signal peptidase. The newly synthesized portions then traverse the major secretory route in the cell: rough endoplasmic reticulum, Golgi apparatus, secretory vesicles, prior to entering the plasma, during which process they are subject to various post-translational modifications like proteolysis, glycosylation, phosphorylation, etc. The transit time through the hepatocyte from the site of synthesis to plasma vary from 30 minutes to several hours for individual proteins. So that's the diagrammatic representation of, you can see the proteins into the lumen of the endoplasmic reticulum, and these are finally released into the bloodstream. Most plasma proteins are covalently modified by addition of either N or O-linked oligosaccharide chains or both. Albumin is the major exception. These oligosaccharide chains perform various functions. Loss of terminal sialic acid residues accelerates clearance of plasma glycoproteins from the circulation. A polymorphism is a Mendelian or monogenic trait that exists in the population in at least two phenotypes, neither of which is rare. ABO blood group systems are best examples of human polymorphisms. Other plasma proteins which exhibit polymorphisms include alpha 1 antitrypsin, haptoglobin, transferrin, ceruloplasmin, and immunoglobulins. The half-life of a plasma protein is the time required for 50% of the molecules present at any given moment to be degraded or otherwise cleared from the blood. Under normal circumstances, the resulting turnover or replacement of older protein molecules with newly synthesized ones occurs without any change in their total concentration. In other words, the countervailing processes of synthesis and clearance reach a steady state where no net increase or decrease is evident at gross macromolecular level. So, half-life may be decreased in diseases like Crohn's disease or protein-losing enteropathies. The aggregate concentration of proteins present in plasma typically falls in the range of 7 to 7.5 grams per deciliter for humans. The osmotic pressure or oncotic pressure exerted by plasma proteins is approximately 25 mmHg. Since the hydrostatic pressure in arteriole is approximately 37 mmHg, with an interstitial tissue pressure of 1 mmHg opposing it, a net outward force of 11 mmHg drives the fluid into the interstitial space. In venules, the hydrostatic pressure is 17 mmHg, oncotic pressure 25 mmHg, interstitial pressure 1 mmHg. A net force of 9 mm attracts water back into the circulation. The above pressures are referred to as the Starling forces. If concentration of plasma proteins is markedly diminished, for example due to severe protein malnutrition, fluid will no longer flow back into intravascular compartment. The resulting accumulation of fluid in extravascular tissue spaces results in a condition known as edema. So here the answer is A. Plasma proteins may act as antiproteinases. These may play part in blood coagulation. Certain plasma proteins act as enzymes. Certain plasma proteins have hormonal functions. Then they play an important part in immune defense in the form of immunoglobulins, complement proteins, and beta 2 macroglobulin. They have involvement in the inflammatory response, especially the acute phase proteins, C reactive protein, and alpha 1 acid glycoprotein. Oncofetal protein may act as a marker, may be used as a marker. Then they have important transport functions. Liver synthesizes 12 grams of albumin per day, representing 25% of hepatic protein synthesis and half of its secreted protein. About 40% of body's albumin circulates in plasma, where it accounts for roughly 3/5 of total plasma protein by weight. The remainder resides in extracellular space. Because of its relatively low molecular mass, about 69 kilodaltons, and high concentration, it is thought to be responsible for 75 to 80% of osmotic pressure of human plasma. Like most other secreted proteins, albumin is initially secreted as pre-protein. Its signal peptide is removed as it passes into cisternae of rough endoplasmic reticulum, and a hexapeptide at the resulting amino acid terminal is subsequently cleaved off further along the secretory pathway. Mature human albumin consists of single polypeptide of 585 amino acids in length that is organized into three functional domains. Its ellipsoidal conformation is stabilized by a total of 17 intra-chain disulfide bonds. One of the major roles of albumin is to bind and thereby facilitate the transport of numerous ligands. These include free fatty acids, calcium, certain steroid hormones, bilirubin, copper, and tryptophan. A variety of drugs including sulfonamide, penicillin G, dicumarol, aspirin are also transported by albumin. This has important pharmacologic implications. Preparations of human albumin have been widely used in the treatment of burns and hemorrhagic shock. Certain humans suffer from genetic mutations which impair their ability to synthesize albumin. Individuals whose plasma is completely devoid of albumin are said to exhibit albuminemia. Although albumin is normally the major determinant of plasma osmotic pressure, persons suffering from albuminemia show only moderate edema. The plasma of patients with liver disease often shows a decrease in ratio of albumin to globulin, decreased albumin globulin ratio. The synthesis of albumin decreases relatively early in conditions of protein malnutrition, such as kwashiorkor. So here the answer is A. These are the various causes of the hypoalbuminemia. Albumin fatty acid complex cannot cross the blood-brain barrier. Albumin binds bilirubin, and it is also involved in certain drug interactions. So these are the various questions related to the plasma proteins. Alpha-1 antitrypsin is also a plasma protein, and that's a problem-based question based on the alpha-1 antitrypsin. Pre-albumin moves ahead of albumin on electrophoresis. It is also called transthyretin. It is involved in transport. Globulins comprise 29 to 54% of plasma proteins, and these are separated by electrophoresis into four fractions: alpha 1, alpha 2, beta, and gamma. Gamma globulins are the antibodies. All globulins are glycoproteins. So here you can see that the plasma proteins which are transporting the other ligands are sitting in the front portion of this hypothetical truck, and the ligands which are being transported by these proteins are sitting in the back trolley of this truck. So these are the transport functions for albumin. These are the various drugs transported by albumin. So these are the ligands transported by the cortisol binding globulin. Now the transport functions of the haptoglobin. Hemopexin binds heme and forms heme hemopexin complex, which is removed by the parenchymal cells, and heme is released back into the circulation. Transferrin is involved in the transport of iron. Alpha-1 lipoproteins are large assemblies of lipids and proteins. The levels of acute phase proteins may increase from 50% to as much as 1000-fold. For example, in the case of C-reactive protein, during chronic inflammatory states and in patients with cancer. Interleukin-1 is a polypeptide released from mononuclear phagocytic cells. It is the principal but not the sole stimulator of the synthesis of the majority of acute phase reactants by hepatocytes, additional molecules like interleukin 6 are also involved. C-reactive protein is so named because it reacts with the C polysaccharide of pneumococci. Alpha-1 antiproteinase, heptoglobin, alpha-1 glycoprotein, and fibrinogen are classified as acute phase proteins. C-reactive protein can stimulate classic complement pathway. Alpha-1 antitrypsin neutralizes certain proteases released during acute inflammatory state. So that is the C-reactive protein. It stimulates the complement pathway. Ceruloplasmin is a copper-containing plasma protein synthesized by liver. It is a ferrioxidase required for oxidation of ferrous to ferric. It is mainly synthesized in the liver. It is also involved in the transport of copper. Low levels of ceruloplasmin are seen in Wilson's disease, malnutrition, nephrosis, and cirrhosis. High levels are seen in acute hepatitis, biliary cirrhosis, hemochromatosis, and obstructive biliary disease. So in Wilson's disease, there is mutation in the gene encoding copper binding ATPase in cells, and copper is not excreted in bile. There is copper toxicity and leading to the hepatocellular degeneration and cirrhosis. It also affects the basal ganglia, and there is a Kesher-Fisher ring seen in the cornea. Alpha-1 acid glycoprotein is a reliable indicator of inflammation. It is increased in acute and chronic inflammatory diseases, whereas it may be increased or decreased in malignant conditions. Alpha-1 globulin is present in high concentrations during pregnancy, but it may also be increased in hepatocellular carcinoma and teratoma, so it may be used as a marker for disease. Alpha-2 macroglobulin is a main component of the alpha-2 globulins, and it inactivates all proteases. Alpha-1 antitrypsin is also called alpha antiprotease inhibitor, and it inhibits all the proteases. ATT deficiency may be caused in protein losing disorders, or functional inactivation of alpha-1 antitrypsin may occur in the smokers due to the oxidation of methionine residues in the alpha-1 antitrypsin. Alpha-1 antitrypsin is increased in inflammation, and it is also a tumor marker for germ cell tumors. For beta globulins, the predominant fractions are hemopexin and transferrin, and the beta lipoproteins. Beta lipoproteins are involved in the transport of lipids. Beta 2 macroglobulin and chiroglobulins also are present in the beta fractions. Chiroglobulins are increased in rheumatoid arthritis, lymphocytic leukemia, multiple myeloma, lymphosarcoma, and SLE.